Somewhere off the coast of Virginia, in the middle of the Chesapeake Bay, there is an island where the surnames have not changed in 300 years. Crockett, Pruitt, Parks, Dyes. The watermen who work the crab boats out of Tangier Island carry the same last names as the men who first cleared its marsh grass in the 1680s. And when geneticists finally ran the numbers on this place, what they found was not a surprise so much as a confirmation of something the island had always known about itself. The DNA had not moved. Everyone else in America had scattered west and south and into cities that did not exist when their ancestors landed, but a small number of families had simply stayed. Not stayed close, stayed exactly. Same peninsula, same hollow, same island, generation after generation, while 40 generations of everyone else's ancestors packed a wagon and kept going. This is a story about what happens to a bloodline when it refuses to travel. Roughly 50 million Americans can trace some documented line back to a colonial-era settler, someone who arrived before the Revolution, before the country had a name of its own. But documentation is not the same as biology, and biology is not the same as geography. Most of those 50 million are descended from ancestors who came, then moved again, mixing with new arrivals at every stop until the original genetic signal thinned into something almost unrecoverable. A tiny fraction did something different. They stayed put. If you're enjoying this story so far, don't forget to hit the like button and subscribe. Drop a comment telling me where in the world you're watching from. I love hearing from you. When researchers compared these families against the broader American population, against genetic databases stretching back to the first English, Scottish, and African arrivals of the 1600s, a small number of isolated communities returned results that looked less like modern America and more like a sealed time capsule, a population that had been marrying within a few square miles of itself since before the United States existed. Jamestown was founded in 1607 on a swampy, malarial peninsula that the colonists themselves nearly abandoned twice in the first 3 years. Roughly 6,000 people crossed the Atlantic to that settlement and its surrounding plantations in the first 2 decades, and by some estimates, 4 out of 5 of them died before ever seeing a second harvest. The ones who survived became the foundation of what genealogists later called the First Families of Virginia, a designation that sounds grander than the reality, which was mostly indentured servants, minor gentry, and a handful of ambitious younger sons who had no inheritance waiting for them back in England. 13 years later, in 1620, a smaller and better documented group crossed on a single ship to Plymouth, in a colony that would eventually produce a genealogical industry of its own, the General Society of Mayflower Descendants, which today counts membership applications from over 100,000 living Americans who can produce a paper trail back to one of the roughly 102 passengers on that crossing. Further south, in the 1670s and 80s, English settlers began arriving along the Carolina coast, drawn by land grants and the promise of a warmer growing season, founding a string of port towns whose churchyards still hold headstones with dates that predate the country by a full century. Paper trails, though, are not blood. A family Bible with a name in it proves that someone believed they were related to a certain ancestor. It does not prove the biology held. Between 1620 and today, 4 centuries have passed, and across 4 centuries, most American families intermarried, migrated, adopted children informally, and in some documented cases, simply invented a connection to a prestigious colonial name because the social capital was worth more than the truth. Genealogists have known this for decades. What they did not have, until relatively recently, was a way to check the biology against the paperwork at scale. That changed with the arrival of large commercial DNA databases and, more importantly, with the academic projects that used isolated, well-documented American populations as natural laboratories for tracking exactly how much genetic material survives, undiluted, across 400 years. Tangier Island is the case study every genealogical geneticist eventually gets asked about, because it is close to a perfect closed system. The island sits roughly 12 miles from the nearest point on the Virginia mainland, and for most of its history it was reachable only by boat, in weather that did not always cooperate. English settlers, largely from the West Country, arrived in the late 1600s. For the next 250 years, the island's population grew almost entirely through births rather than new arrivals, which produced 2 well-documented effects. The first is linguistic. Tangier's watermen still speak with a dialect that outside researchers have described as one of the closest surviving relatives of Restoration-era English spoken anywhere in the United States, a claim that gets exaggerated in casual conversation, but that linguists studying the island's vowel patterns take seriously enough to keep publishing on it. The second effect is genetic, and it is the less romantic, more clinical twin of the dialect story. When researchers sampled Y chromosome markers from Tangier's long resident surnames, the same handful of English paternal lines kept appearing, generation after generation, with almost none of the outside admixture you would expect to find in a mainland Virginia population over the same period. The island had not been isolated because anyone wanted a genetic experiment. It had been isolated because the water was 12 miles wide, and a crab boat is not a fast way to meet someone from Baltimore. Tangier is not unique, only unusually well studied. Up and down the eastern seaboard and deep into the Appalachian interior, similar pockets exist, places where the same small set of colonial-era surnames has persisted with minimal outside mixing since before independence. Ocracoke, on North Carolina's Outer Banks, tells almost the identical story. An island population descended largely from English and a smaller number of Scottish settlers who arrived in the early 1700s and then found themselves, like Tangier, cut off by water and weather for the better part of 3 centuries. Linguists call the local accent the Ocracoke brogue, and it carries vowel sounds researchers have connected to 17th and 18th century English dialects that have otherwise vanished, even in England. The Appalachian interior offers a different mechanism for the same outcome. It was not water that isolated these communities, but topography, narrow hollows and steep ridgelines that made travel to the next valley a genuine undertaking well into the 20th century, in some counties until the highway projects of the 1950s finally cut through. Families in these hollows married within a radius of a few miles for generation after generation, not out of any deliberate policy, but because the physical geography made a wider search for a spouse impractical. Surname studies from these counties show the same clustering effect as the island communities. A small founding set of colonial-era names repeated and interlinked across 24 generations, with genetic markers that track back cleanly to specific counties in England, Scotland, and the north of Ireland. Some of these communities, particularly in the Cumberland Plateau region of eastern Tennessee and Kentucky, have been studied for a related but distinct reason: a set of visible traits and rare genetic conditions, including a documented cluster of methemoglobinemia that appeared far more frequently in these populations than in the country as a whole, a direct and measurable consequence of generations of marriage within an unusually small founding gene pool. What the researchers are actually measuring when they run these comparisons is called identity by descent, a technical term for shared DNA segments that 2 people inherited from the same specific ancestor rather than from the broader human gene pool. In a normal, highly mixed American population, identity by descent segments from a single 17th century ancestor have usually been diluted past the point of reliable detection by the time you reach a 10th or 12th generation descendant. Too many other ancestors have been added to the family tree in the meantime, each contributing their own share of DNA and pushing the original signal toward statistical noise. In an isolated population like Tangier or the more insular Appalachian counties, that dilution slows dramatically because the pool of available ancestors stays small. If your grandparents, and their grandparents before them, were all drawing from the same few hundred people on the same island or in the same 3 connected hollows, the original colonial-era genetic signal does not get diluted so much as it gets recirculated. The same core DNA keeps reappearing, generation after generation, concentrated rather than scattered. There is a second category worth understanding here, distinct from the island and hollow populations, and it involves land rather than water or mountains. Certain Tidewater, Virginia and low-country South Carolina families held the same plantation acreage, under the same surname, from the late 1600s through the Civil War, and, in a smaller number of documented cases, into the present day. Land continuity is not the same as genetic continuity, and researchers are careful about the distinction, because a family can hold the same deed for 3 centuries while marrying widely and diversely the entire time. But in a meaningful number of these long-tenured landholding families, particularly ones in rural counties with limited outside migration before the 20th century, genealogists have found both forms of continuity together, the same land and a traceable, largely unbroken paternal or maternal line. One frequently cited case involves a family holding land in Surry County, Virginia, continuously since the 1630s, with documentation, church registers, tax rolls, and probate records connecting 11 consecutive generations to the same few hundred acres along the James River. When a Y-DNA sample from a living male descendant was compared against historical church burial records and cross-referenced with a distant cousin's line that had split off and moved to Kentucky in the early 1700s, the markers matched with a precision that genealogists in this field describe, without exaggeration, as remarkable for a family separated by nearly 300 years and a full continent's width of geography. New England offers a comparable, if less dramatic, example. Certain families in Plymouth and Barnstable counties in Massachusetts, descended from Mayflower passengers, remained on Cape Cod and the South Shore for a full 10 generations before the first documented outward migration in family records, a pattern researchers attribute partly to the peninsula's relative isolation and partly to a fishing and shipbuilding economy that gave sons a reason to stay rather than push west with the rest of the colony. The Melungeon communities of the Cumberland Plateau add a further layer to this picture, and for a long time they were treated less as a genealogical case study than as a regional mystery, a population of mixed European, African, and indigenous ancestry that had settled the ridges of eastern Tennessee, southwestern Virginia, and eastern Kentucky by the late 1700s and remained there, largely intermarrying within the same network of families, for the next 2 centuries. 19th century outsiders wrote about the Melungeons with a mixture of curiosity and suspicion, inventing origin stories that ranged from shipwrecked Portuguese sailors to lost Roanoke colonists, mostly because the actual, more mundane explanation, a community of free people of color and their European neighbors settling remote land where county officials rarely asked questions about ancestry, did not fit the folklore outsiders preferred. When genetic researchers finally sampled surnames long associated with Melungeon families in the 2000s, the results supported the less romantic explanation. The Y chromosome and mitochondrial markers matched a combination of sub-Saharan African, Western European, and in a smaller number of lines, indigenous American ancestry, consistent with free families of mixed heritage who settled a rugged, thinly governed stretch of frontier in the middle of the 1700s, and then, like Tangier's watermen a few hundred miles east, simply stayed for 200 years while the rest of the country kept moving around them. Pennsylvania's German-speaking communities offer a different kind of closed population, this one preserved by religious practice rather than water, mountain, or frontier isolation. Old Order Amish and conservative Mennonite communities in Lancaster County trace almost entirely to a founding population of fewer than 500 individuals who arrived from the Rhine Valley and Switzerland in the early 1700s, and because these communities have maintained near total endogamy for 3 centuries as a matter of religious discipline rather than geography, their genetic record is in some ways the cleanest of any population discussed here. Medical geneticists have studied Lancaster County Amish extensively, not primarily for genealogical interest, but because the same small founding population that preserved a readable 17th century genetic signal also concentrated a handful of rare recessive conditions to rates far higher than the general population, a direct and well-documented consequence of 300 years spent marrying almost exclusively within the same few hundred founding surnames. Coastal Maine offers one more variation worth naming before moving on. Quieter than Tangier's story, but built from the same basic ingredients. Fishing villages along the state's mid-coast, places like Monhegan Island and stretches of the down east shore, were settled by English families in the late 1600s and early 1700s, and then left largely alone by the waves of immigration that reshaped most of the rest of the Northeast across the 1800s. A hard coastline, a short growing season, and an economy built entirely around the sea gave these communities little reason to attract new arrivals, and for long stretches, little practical way to leave. Surname records from island parishes in this region show the same narrow, repeating founding set as the Chesapeake and Outer Banks communities, a small cluster of English names recurring across 24 generations with only occasional new blood entering through marriage to a neighboring village's equally narrow founding pool. If you're hooked on this story, make sure to subscribe and turn on notifications so you never miss an upload. Your support means everything and helps me bring you more emotional stories like this one. The contrast with the rest of the country's colonial descended population is where this story earns its edge. The average American who can document an ancestor from the 1600s is not the descendant of someone who stayed. They are the descendant of someone who left, repeatedly, for 3 or 4 centuries running. The colonial coast emptied westward through the Cumberland Gap in the late 1700s, a single mountain pass that funneled an estimated 200,000 settlers into Kentucky and Tennessee in barely 2 decades. It emptied again onto the Ohio frontier in the early 1800s, again into Texas and the Mississippi Valley before the Civil War, and again onto the Great Plains after it, and once more into California and the Pacific Northwest by the century's end. Each migration wave mixed the traveling families with new populations, immigrants from Germany, Ireland, Scandinavia, and later from nearly every part of the world, and each mixing event diluted the original colonial genetic signal a little further. By the time a family has moved 4 or 5 times across 4 centuries and married outside the original founding population at each stop, the amount of identifiable 17th century DNA remaining in a living descendant can shrink to a fraction of 1%, present in the genealogical paperwork but nearly invisible in the genome itself. This is not a flaw in the science. It is simply what 400 years of American mobility does to a bloodline. The families that never left are, in a strict genetic sense, the outliers, the control group against which the rest of the country's colonial ancestry claims can be measured. And what they reveal, when researchers line them up against the broader population, is a quiet correction to a story most descended families like to tell about themselves. A name on a family tree connecting you to a Mayflower passenger or a Jamestown settler is a real historical claim, supported by real documents, and there is nothing false about it. But it is a claim about paperwork and memory. The biology, in most cases, has been diluted across 300 and some years of American restlessness into something a genome cannot reliably confirm on its own. Only in the places where the restlessness never happened, the islands 12 miles offshore, the hollows behind the ridgeline, the plantation acreage that stayed in one name for 11 generations, does the DNA still carry an unbroken signal loud enough to read. There is a further wrinkle that surprises even genealogists who specialize in this work, and it concerns Africa rather than England or Scotland. The transatlantic slave trade brought roughly 400,000 documented Africans directly to what is now the United States, between the 1600s and the legal closing of the trade in 1808, and a significant number of Black American families, particularly in rural counties of the Low Country, the Virginia Tidewater, and parts of the Mississippi Delta, show the same isolation pattern as the White Island and Hollow communities, for reasons rooted in a very different and much harsher history. Enslaved and later freed Black families in isolated rural counties frequently remained in the same narrow geography for generations, sometimes on the same land their ancestors had been forced to work, with limited opportunity, or, for long periods, legal ability to relocate freely. Geneticists studying Gullah Geechee communities along the coast of South Carolina and Georgia have documented some of the clearest identity by descent signals connecting living residents to specific West African regions anywhere in the African diaspora, a direct result of the same geographic isolation, marshland, barrier islands, and limited road access that preserved Tangier's English dialect a few hundred miles up the coast. Two entirely different histories, one shaped by choice and the other by force, produced a strikingly similar genetic outcome because the underlying mechanism, geographic isolation slowing the normal dilution of a founding population's DNA, works the same way regardless of why the isolation happened in the first place. Louisiana offers yet another variation on the same theme, this one tied to language rather than water or ridgeline. Acadian families expelled from Nova Scotia by the British in the 1750s resettled along the bayous of southern Louisiana, and for the better part of 2 centuries afterward, Cajun communities in parishes like Lafourche and Terrebonne married almost exclusively within a network of related founding families, preserving both a distinct dialect of French and a genetic signature that researchers can trace with unusual precision back to a small number of specific parishes in western France by way of a single, brutal deportation. The isolation here was partly geographic. The bayou country was genuinely difficult to reach for outsiders well into the 20th century, and partly social, a shared history of displacement that bound these communities together long after the original expulsion had faded from living memory. None of this genealogical work would be possible at scale without the specific method researchers use to date a shared ancestor from the length of a shared DNA segment. Every time a chromosome passes from parent to child, it gets reshuffled through a process called recombination, which breaks the original inherited segments into shorter and shorter pieces with each passing generation. A segment shared with a first cousin is long, easy to spot, and unmistakable. A segment shared with a descendant of the same ancestor from 12 generations back is short, fragmented, and easy to lose in statistical noise unless the surrounding population has stayed small enough that the same handful of ancestral segments keep reappearing rather than being diluted into invisibility. This is the precise mechanism that makes island, hollow, and closed community populations so valuable to researchers. It is not that their ancestors were somehow more important than anyone else's. It is that the mathematics of recombination had far fewer places to hide the signal. Commercial genealogy companies complicated this picture when they entered the market in the 2000s, because their reference panels were built primarily from modern, highly mixed populations rather than from the specific closed communities discussed here. A Tangier waterman who takes a mainstream consumer DNA test will often see an ancestry breakdown dominated by a generic English or British Isles category, the same result a recently arrived immigrant from London might receive, because the algorithm is comparing his DNA to a broad modern reference population rather than to the specific, much narrower 17th century Chesapeake founding population his family actually descends from. Academic researchers working directly with these communities use narrower, custom reference panels built from historical church and parish records specific to the exact region a family's documented ancestors left from, and the results those narrower panels produce are considerably more precise and considerably more revealing than anything a general consumer test can offer. This is also why claims made in this kind of research need to be read carefully. A study can demonstrate that a Tangier family's Y chromosome markers cluster tightly with historical West Country English populations. It cannot, and should not, claim to identify a specific named ancestor from a specific ship's manifest without the paper record to support it. The strongest work in this field treats the DNA and the documentary record as two separate lines of evidence that either confirm each other or sometimes quietly do not, and either outcome is treated as data rather than disappointment. Genealogical societies have started paying closer attention to this distinction over the past decade, partly because DNA testing has occasionally overturned a family's long-held paper genealogy entirely. A documented paper trail connecting a family to a prominent colonial name can, and sometimes does, turn out to run through a 19th century informal adoption, a second marriage that was never fully recorded, or a simple clerical error in a county courthouse that got copied forward for 150 years before anyone thought to question it. In the closed, isolated populations this story has focused on, that kind of quiet correction is rarer, not because the record-keeping was better, but because the small size of the founding population left fewer alternative explanations available. If nearly everyone on a 12-mile island descends from the same 40 or 50 founding households, there are only so many places a DNA result can plausibly lead, and the paper trail and the genetic trail tend, more often than not, to arrive at the same place. What all of this adds up to across islands, hollows, plantation acreage, Cajun bayou parishes, Melungeon ridgelines, and Pennsylvania farmland is a small and shrinking set of American places where the deep past is not a matter of inference but of direct, testable biological continuity. These are not the families history books tend to celebrate. They rarely produced founding fathers or famous generals. What they produced instead, patiently and without any particular sense of occasion, was an unbroken chain of ordinary births in the same small stretch of geography for 300 or 400 years running, a kind of continuity that the rest of the country's more storied and more mobile colonial descendants, for all their famous names and documented pedigrees, generally cannot claim at the level of the genome itself. None of this means an American with a well-mixed, well-traveled family tree has a lesser or less authentic connection to their colonial ancestors. Genealogy has never claimed to be only about DNA, and the paper record, the wills, the church registers, the census entries, the family Bibles passed down through 6 or 7 households remains a legitimate and often more complete account of where a family actually came from than a DNA test can offer on its own. What the isolated populations demonstrate is something narrower and, in its own way, more interesting. They show what an American bloodline looks like when the great national habit of moving on gets interrupted. The country's entire identity, in some sense, is built on the idea of leaving, the frontier, the westward expansion, the next town over, promising a better plot of land. The families who never left are the exception that makes the rule visible. Their DNA did not thin out because the ancestors who founded those island and hollow populations were, geographically speaking, the ones who stayed home while the rest of the country kept walking. Researchers who work in this field describe visiting these communities with a kind of professional caution, aware that a small, tightly interrelated population is not just a genetic curiosity but a living place with its own concerns about privacy, about outside attention, and in Tangier's case, about a shrinking population and eroding shoreline that may leave the island uninhabitable within a few decades, regardless of what its genome reveals. The Chesapeake has claimed roughly 2/3 of the island's original landmass since the 1800s, and current population estimates put year-round residents at under 500, down from over 1,000 at the community's mid-20th century peak. If Tangier disappears as a place people actually live, the unbroken genetic line that took 300 years to preserve will not vanish from the historical record. The DNA samples already collected will remain, but the living, breathing continuation of that line, the version still walking around on a crab boat instead of existing only in a database, will end. The same pressure, in slower and less dramatic forms, is working on the Appalachian hollow communities. the Ocracoke Brogue speakers, and the remaining Gullah Geechee settlements, as roads improve, as young people leave for work in ways their great-grandparents structurally could not, and as the isolation that preserved these bloodlines for 4 centuries finally, quietly, comes to an end. Genealogists who track this kind of population data describe a narrowing window, perhaps 2 or 3 more generations, before the last of these closed founding populations dissolve into the broader, more mobile national gene pool, the way nearly every other American family already has. What remains, for now, is a genetic record unlike almost anything else in the country's family history. A living population whose ancestors watched the entire rest of America pack up and move west, again and again, and chose, generation after generation, without ever framing it as a choice, to stay exactly where they were. Their surnames have not changed. Their church registers, in some cases, stretch back an unbroken 300 years without a gap. And when a geneticist finally ran their chromosomes against the broader American population, what came back was not a mystery so much as a confirmation of the thing everyone on that 12-mile stretch of water had always quietly understood about themselves. The rest of the country's ancestors kept moving. Theirs never did. 400 years from the first landing at Jamestown, a small number of American families are still standing, more or less, on the exact ground where their story began, carrying a genetic record the rest of the country lost somewhere on the road west. It is worth sitting with what that actually represents. Every other line of a family tree in this country, at some point, made a decision to leave. Whether that decision was a wagon axle pointed toward Kentucky, a train ticket bought in a Pennsylvania rail yard, or a much older and far crueler decision made for someone else entirely. The handful of places where that decision never got made are not more American for having stayed. They are simply the control group, the quiet baseline against which the rest of the country's restlessness can finally be measured in the language of chromosomes rather than family legend. A crab boat idling off a marsh at dawn, a church register with 3 unbroken centuries of the same handful of names, a hollow where the road did not arrive until the middle of the last century, these are not dramatic images on their own. But set against the vast, restless churn of American migration, they become something closer to an anchor point, the rare place where the past did not need to be reconstructed from paperwork because it had simply never left the room. Thank you so much for watching until the end. If you loved this story, check out the other videos on your screen now. I think you'll really enjoy them. And don't forget to subscribe if you haven't already. See you in the next story.